All right, so we've done carbohydrates, we've done lipids. We're now going to jump forward. The book goes to proteins next, but I'm going to go to nucleic acids. It makes more sense this way in terms of how the information in our genomes is kind of expressed, and we'll come back to this in greater detail. But we're going to talk as an overview over the group of molecules we call nucleic acids. There are two primary nucleic acids here. We'll talk about both of them and a little bit about their structure here. Their structure is really important to how they work. We'll see that later in coming chapters in the genetics and stuff like that. And then we'll go into a little bit of detail about what role they play in the cell as well. So we have two nucleic acids. We have ribonucleic acid, RNA, and deoxyribonucleic acid, which is DNA. The DNA is our genetic storage information. That's where our genetic information is stored. It's kind of like the blueprints. The blueprints for a house don't do anything, but they give all the instructions for things that need to be built. So we can read the blueprints and then go make things from them. RNA, on the other hand, has several diverse functions. It is primarily involved in the process of what we call gene expression. Gene expression is turning genes on and off. I will define precisely what a gene is, and the definition of a gene is very particular for this course. If you Google the definition of a gene, you will get several definitions. They are not necessarily all wrong, but there is a correct context that we're going to use the definition of a gene in for this course. These two molecules are actually quite similar. They differ in what sugar is in there, and for the most part, DNA is double-stranded, so it's got these two backbones, and in between are kind of these rungs like on a ladder, and they are made up of nucleotides. The nucleotides in DNA are A, T, C, and G. We'll talk more about them later and how they pair up and things like that. RNA, on the other hand, is often single-stranded. It can be double-stranded. There are some cases where it can be double-stranded, but it is most often single-stranded. And for whatever reason, evolution happens and doesn't always make sense. RNA uses the bases A, U, C, and G. U operates pretty much similarly to T in DNA. It's just a slightly different chemical. That's just the way it works. We'll talk more about RNA and the different processes it can be involved in. But DNA is the storage material. RNA is involved in gene expression, turning genes on and off. So let's do a couple of critical definitions here, because you probably have heard these words, but you might not have a specific definition, and we need to know those specific definitions for this course. We have two terms, genome and gene. So, the genome. You've probably heard of the Human Genome Project. Okay, when I was in high school, they were working furiously on sequencing the human genome, and the genome is all of the DNA contained within a cell. So you have a cell, and the genome is all of the DNA that's in there. Okay. We have an area of study that's called genomics that studies genomes, and included in the genome are segments that we call genes. I will define those in a second. The genome is contained within a chromosome or chromosomes. In prokaryotes, we generally have a single circular chromosome. There might be extra bits in there, but we have the single circular chromosome, and there's one of those. In eukaryotes, we tend to have many different linear chromosomes. You might be familiar with this. This is a human karyotype. We have 23 different types of chromosomes that all of our DNA is stored in. We'll come back to chromosomes when we get into genetics. So that's the genome, all of the DNA contained within a cell. Now, Not all of the DNA is what we call genes. A gene is a sequence of DNA that codes for a transcript. I'm going to say this a different way, but it's the same thing. A sequence of DNA that codes for an RNA. So it's a little region of the DNA that can be turned into an RNA. Most genes code for mRNA. They will get turned into mRNA, which will then get turned into a protein. So we used to say that genes were areas that coded for proteins, but that's not quite true. Our definition is going to be really specific and very modern, a sequence of DNA that codes for a transcript or an RNA. That is what a gene is. Okay, we will learn more about that. Please memorize that definition now. It will make more sense later, but do not go to Google and search the definition of a gene, because I will ask you to define a gene on the study guide. If you go to Google to look for that, you will find that their definition is something like a unit of heredity. While that's not technically incorrect, this is a more precise definition, and this is the definition we will use for the course. Okay. That's our two terminologies that we need for right now. What are these polymers made up of? Well, deoxyribonucleic acid, DNA, has nucleotides with the sugar deoxyribose in it. Ribonucleic acid, RNA, has ribose in its nucleotides. A nucleotide is our monomer that makes up our nucleic acid. We have three parts to these. We have the sugar, that's either deoxyribose or ribose. We have the phosphate group. This will be important in DNA replication. And we have what's called the nitrogenous base. The nitrogenous base is the information containing part. So all nucleotides are the same over here. All DNA ones are the same and all RNA ones are the same. But what varies is the nitrogenous base. Now I just want to point out that you might see 5' carbon, 3' carbon. That will be important later. I just want to show you that organic chemists, they have to label things. So there's a 1' carbon here, 2' carbon, 3' carbon, 4' carbon, 5' carbon. They've labeled them so we know which one we're talking about. That'll be important because later we'll see that new DNA nucleotides can only be added onto this 3' end. So DNA is going to be built in a very specific way. All right, let's talk about the information containing bit, the nitrogenous base. These are the ATs, Gs, and Cs that you might be familiar with. You don't need to memorize their structure or even really their name, although it's good to have that in your memory. But A is adenine, C is cytosine, G is guanine, T is thymine. Thymine is found in DNA, but in RNA we have this uracil, U, that is found. And so U and T are, they function the same, but one is found in DNA and one is found in RNA. Sorry, that's just the way the system evolved. We will see that when nucleotides pair up, we have A that pairs with T and G that pairs with C. Let's talk about why that is. So we're going back to our chemistry. Here is our DNA double helix 3D model. We have the backbones here. You can see the phosphorus. Remember, that's part of the nucleotide. And then in the middle are the nitrogenous bases. So if we zoom in here, we have A paired with T and G paired with C here. Okay. What actually holds these together? I've mentioned it a few times. Look at this structure. Think about what I might have mentioned previously. Pause the video if you need to. Okay, so we've talked about a couple of different types of bonds, and I mentioned that DNA is held together, these nucleotides are held together with hydrogen bonds. Those hydrogen bonds are relatively weak. We can break them to open the DNA up. And these kind of sides here are held together by strong covalent bonds. So We will not break the DNA when we unzip it. We'll just kind of pull it apart. That's going to allow us to replicate DNA. So hydrogen bonds hold the bases together. Between A and T, there's 2 of them. Between G and C, there's 3 of them. And then the backbone, as it's called, is held together by covalent bonds. They're technically called phosphodiester covalent bonds. That's right here. It holds them together. What I want you to take from this is that there is a direction to them. They go in a 5' to 3' direction. So this one goes downwards and this one goes upwards. That will be important. This middle bit here, we call these bases complementary to one another. So here's one nucleotide, here's the other nucleotide. Because they are A and T, they are complementary to one another and they will base pair together. G and C are complementary together and they will base pair together. The story about the proposal of this double helical structure is cool. You may have heard a little bit about it. You probably have heard of these 2 individuals. I believe this is Watson and this is Crick right here. You've probably heard it said that they discovered the structure of DNA. They didn't really actually do any experiments. They proposed a model, and it was actually based on a lot of other people's data and evidence and chemistry rules and things like that. The story of this is complicated. We'll talk a little bit about the structure in chapter 14. If you want to learn more, I'll tell you a course that talks all about that in a moment. But let's look at the structure here. I just want to reiterate that these bases pair together, but the directions go in opposite directions. So we call these chains anti-parallel. They run in opposite directions. We will come back to this in chapter 14 when we do the structure of DNA. Okay, so if you want to hear more about the model that Crick and Watson came up with, I recommend taking my Micro 250 class. We spend a lot of time talking about that, looking at the evidence that they used, whose experiments, whether they had permission to do that, and so on and so forth. So if you think DNA is super cool, Micro 250 is a great place to do that, learn about DNA. All right, so we know a little bit about the structure. We know that there's DNA and that there's RNA. How does genetic information flow? So I said DNA is the storage material. It's like the blueprints. That means we need to get the information out of the blueprints and into something physical that is being made. This is where what we call the central dogma of molecular biology comes in. This is our flow of information. This is how genetic information flows. We have the DNA here, and in the DNA are regions of DNA that we call genes. Genes code for a transcript for an RNA. So we make RNA through a process called transcription. Using the information from DNA, we make an RNA through transcription. That RNA goes out and it's going to be translated by a structure called a ribosome. The ribosome reads the RNA and turns it into a protein. Now I should say, this happens for mRNA. There are actually several different types of RNA. I'm going to show you those in a sec, but let's just go back through this. DNA, the genes get converted by transcription into RNA. Those mRNAs get translated into proteins, and the proteins go out and do things. Here's my GFP protein. It will go and fluoresce green. All right, let's talk about the different types of RNA, because I've been kind of specific here, right? All right, so there's 4 types of RNAs. You probably have heard of mRNA or messenger RNA. We have these things called mRNA vaccines that have been pretty big in the news recently. mRNA is the one that I was just talking about in the central dogma diagram. mRNA takes the message from the DNA to a ribosome, and there it is converted into a protein through a process called translation. We will learn all about that. A ribosome takes the mRNA and translates it into a chain of amino acids, which we call a protein. All right, so that's one type of RNA, but there are others. One of them is called ribosomal RNA, or rRNA. The ribosome is a mixture of this ribosomal RNA and protein, and it works to translate the mRNA. So it's part of the mRNA translation machinery, what we call a ribosome. The ribosome is part rRNA, part protein, and it's going to take the mRNA and translate the message there. Ribosomal RNA is kind of cool because it's actually catalytic. That means it can help reactions occur. That's really interesting because DNA doesn't do that. And there's some hypotheses that propose that probably the earliest genetic material out there was RNA because it's so versatile. It can do so many different things. It can catalyze reactions. It can store genetic information. RNA is pretty cool in that way. Another type of RNA is called transfer RNA, or tRNA. This will be involved in the actual decoding of the mRNA. So here's our ribosome, here's our mRNA, and then we have these things called tRNAs. tRNAs come in and they match up with the mRNA, and they bring with them the amino acids that will get built into a protein chain. So they're kind of helping translate the message. Okay, so all three of these things work together. We then have a side class of RNAs that only has really been discovered in the past 20 years here. And we're still learning about them. We're finding more of them. These are called microRNAs, or miRNAs. These are what we call short non-coding RNAs. They don't code for a gene. They're involved in turning genes on and off. We'll talk more about this later in the course. We need to understand how the genetic information is transcribed and translated first, but microRNAs can regulate gene expression. So those are our four types of RNA. We got the messenger RNA that carries the message that will be converted into a protein. Ribosomal RNA, rRNA, works as part of the ribosome to help translate that message. Transfer RNAs, tRNA, brings the amino acids in to decode the mRNA, and then microRNAs are involved in gene expression. So if you need a handy kind of quick guide between these two things, DNA and RNA, DNA carries the genetic information. RNA is involved in protein synthesis and gene expression. In eukaryotes, DNA is located in the nucleus. It does not leave there. In eukaryotes, RNA is made in the nucleus, but will go on to leave the nucleus. DNA is structured as a double helix, whereas RNA is single-stranded. The sugar in DNA is deoxyribose, that's the D. In RNA, it's ribose. And the bases, they're the same, except RNA has uracil, where DNA has thymine. All right, so here's a review question. You find a single-stranded nucleic acid containing uracil. What type of molecule is it? Okay, go ahead and pause the video. Think about that. All right, hopefully you chose RNA, okay? It's a nucleic acid, so you only had two choices, right? You had DNA or RNA. Those are our two nucleic acids. And here, I've told you that it is single-stranded and contains uracil. That would indicate that it is RNA. Okay. We will talk about what amino acids and polypeptides are in just a second, but they are not nucleic acids. Now let's do another one. Which part of the DNA double helix is stronger? Okay, we've got two options. We got the backbones, which are here, or the rungs of the ladder here. Go ahead and pause the video. Think about that one. All right. Hopefully you chose the backbone is stronger. The backbone is held together by covalent bonds. Those are stronger than the hydrogen bonds, which hold the rungs together in the middle. Okay. So that's our quick overview of nucleic acids. Again, we're just learning the pieces. We're going to put it all together later when we come to the genetics section. So We have our genome. That's all of the DNA in a cell. Genes are regions of DNA that code for a transcript, an RNA. Okay, so they're small pieces of the genome. DNA and RNA are polymers of nucleotides. These are joined together by a phosphodiester bond, and that's the backbone. The double helix middle part is held together by hydrogen bonds. DNA is double-stranded. Its sugar is deoxyribose. A pairs with T, G pairs with C. RNA, on the other hand, is generally single-stranded. Its sugar is ribose. A pairs with U in RNA, and G pairs with C there. We talked about what we call the central dogma of molecular biology. That is, DNA gets transcribed into RNA, mRNA, and that gets translated into protein by the ribosome. There's 4 different types of RNA. We have the messenger RNA that carries the genetic information there. The ribosomal RNA, rRNA, that's part of the ribosome complex. We have transfer RNA, tRNAs, those bring in the amino acids, and then microRNAs are involved in regulating gene expression. Okay, that's our overview of nucleic acids. We're going to move down the line here, and we're going to talk about proteins next, because they're the next step in genetic information here.